Today we will discuss about how the glycocalyx composition affect the cancer progression. So those who are not aware about what this glycocalyx means, this figure on the top left figure shows what this glycocalyx is composed of. Glycocalyx composed of the different glycan structures that are expressed um, on the cell surface or the cell membrane proteins, and they have been categorized into four different categories that are mentioned here. First one is this N-glycans that are usually expressed on the asparagine side of these proteins, and these proteins are since these glycans are expressed on these proteins, they are called glycoproteins. And these O-glycans are usually expressed on serine, either serine or threonine amino acid of the proteins, and or they can also be expressed on the mucins that can be seen here. So these are called as mucins, and other ways by which these glycans are expressed are in the form of these glycolipids, where the glycans are expressed on the lipids, and one of the example is shown here. Next one is this proteoglycans. In this, the uh, long chain glycan structures or glycosamine glycans are present at this serine or threonine sites, and these are, since the number of glycans are more, or these GAGs, glycosamine glycans are expressed on these glycans. These proteins are known as called as these proteoglycans. So this is about the glycocalyx composition and how this change in the glycocalyx compositions will affect this cancer progressions. So we will start with this bottom left figure. So in this figure, what is shown here is that the change in this glycocalyx composition because of the reprogramming that has occurred within the cancer cells. The glycocalyx compositions, the post-translation modification, such as the glycosylation changes because of the reprogramming that has occurred within the cells. And because of the change in the glycosylation, the glycans that are expressed on the cell surface are different from the normal cells, and these different glycan structures or the change in glycocalyx composition will will interact with this actin, and because of this because of the mechanical forces that are transferred of this because of these integrins and these glycocalyx interactions with this ECM, the mechanical force got transferred in the intercellular parts of the cells, and that will trigger uh, intercellular signaling pathways, and uh, and for this actin plays an important role, as these integrins will transfer the force to the actins. Integrins will transfer the force to the actins, and then actin will activate intercellular signaling pathways. And two major signaling pathways that are activated are shown here. One of them is upregulations of these mesochemical genes. Because of the mechanical signaling, it will lead to the upregulation of this mesochemical genes. So these mesochemical genes has the property to change the cell characteristics to from epithelial to migratory, or epithelial to mesochemicals, and cells can now more prone to move from one place to another place. Simultaneously, it will also upregulates this mechanical forces signaling will trigger upregulation of these bulky glycocalyx genes, and these bulky glycocalyx genes will act as a feedback loop, that is, or in a cascade manner, it will make the glycocalyx on the cancer cells more bulkier, and thereby the mechanical force transitions from the integrins to the actins will become more stringent, and this will further enhances or upregulates this downstream signaling pathways to a greater extent, and make the cells more migratory in the nature. So this act as a feedback loop or in a cascade manner for the cancer or tumor cells, when because of the change in the glycocalyx compositions and its interaction with the ECM. So how this can be deal with? There are four different ways are mentioned in this figure, that is on the bottom right hand side. So in this case, first one, we can use these small molecular inhibitors, and these small molecular inhibitors will affect this glycosylation profiles that are leads to this more bulkier uh, glycan compositions of the glycocalyx. And next, we can add this metabolic reprogrammed activated donor. So what it will does is it will reduce the amount of substrate that are available for the transferase, glycotransferase. So the post translation modification or the that uh, glycosylation that usually proceed without this metabolic or the glycosylation will be reduced because of the presence of this activated sugar donor, as the amount of substrate now compete both for the glycosyl transferase and also for this activated donor. So the amount of substrates that are now available for the glycosyl transferase get reduced. Next, they can use these synthetic glycopolymers. In the synthetic glycopolymers, the synthetic glycopolymers are have a higher length. One of the example is shown here, that is 90 nanometer compared to the integrin and the short glycopolymers. And because of the long synthetic glycopolymer, it can interact with the ECM and prevent the interactions of this integrin and short glycopolymers, and thereby downstream signaling pathways will not get activated. So this is another approach, and this synthetic glycopolymers will be formed in a way that it will also not activate any intercellular signaling pathways. Next is or the final panel of this third figure, that is panel D, is talks about this CRISPR-Cas9 based pruning. So in this case, different enzymes uh, that are responsible for this different enzymes that are responsible for the glycosylation process can be knocked down using CRISPR-Cas system. For example, if the MGAT can be knocked down, then the final uh, N-glycan structures will not have this glycnex will not have the sialic acids and galactose. And if this or for the O glycans, if this cos MC is knocked down, then they will not have the sialic acids. And for the glycolipids, if UGCG is knocked down, then there is no glycan structures that are expressed on the lipids. So these things can be used in order to reduce the amount of cancer cells, or in order to reduce the effect of this cascade effect or the glycocalyx compositions on the cancer progression. So I hope you got a basic idea how the glycocalyx composition affect cancer progression. If you need to understand more about their relations and in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.